Hello everyone, welcome to the channel Microbial World. In this video, we will be discussing about cells of the immune system. Our immune system is made up of different immune organs and tissues located all over the body. All these organs are connected by the lymphatic system and the blood vessels into a functional unit. So what is lymphatic system? We know that lymph is a fluid that contains white blood cells, proteins and waste products, right? So the lymphatic system is a network of vessels, nodes and organs that transport lymph throughout the body, okay? White blood cells, that is leukocytes, play a key role in the body's immune responses. So what is the function of these white blood cells? They will defend the body against infectious disease and any foreign material. The cells of the immune system are lymphocytes, which includes T lymphocytes, B lymphocytes and natural killer cells. Phagocytic cells such as monocytes and macrophages. Granulocytic cells such as neutrophils, basophils and eosinophils. Dendritic cells. Lymphoid cells, which is also called lymphocytes. Lymphocytes are small round cells found in the peripheral blood, lymph node, lymphoid organs and in tissues. They constitute 20 to 40% of the body's white blood cells and 99% of cells in the lymph. There are about 10⁻11 lymphocytes in the human body. So these lymphocytes, they circulate continuously in the blood and lymph, and then they will migrate into the body tissue spaces and lymphoid organs, thereby integrating the immune system. Okay. These lymphocytes, they are divided into three populations based on their function and cell membrane components. Okay. So as we said before, they are B lymphocytes, T lymphocytes and natural killer cells. So now let's discuss about B lymphocytes. Bone marrow is the site of maturation of B lymphocytes. Okay. So B lymphocytes arise in the bone marrow through the process of hematopoiesis, and its maturation takes place in the bone marrow itself. Okay. After maturation, they migrate and they express themselves by forming unique antigen binding receptors on their membrane. Okay. So this antigen binding receptor on the membrane of B lymphocyte is called B cell receptors or antibodies. Now, what is the function of B lymphocytes? It is to produce antibodies known as immunoglobulins or gamma globulins. Okay. Migration of mature B cells moves to the bone marrow, lymph node, spleen, some parts of the intestine and the bloodstream. So this is the structure of B lymphocyte. So you can see antigen binding receptor on the membrane of B cell, right? It is called B cell receptor or antibodies, okay? When a naive B cell interacts with an antigen for the first time, this antigen has to match the membrane bound receptors on the B cell, okay? So these B cell receptor or antibodies will bind with the antigen, and this will cause the B cell to divide rapidly, and its progenitor will differentiate into memory B cells and effector B cells known as plasma cells. Okay? And it is the plasma cells that will produce antibodies which are secreted into the bloodstream, tissues, respiratory secretions, intestinal secretions and tears. T lymphocytes or T cells. So these T cells arise in the bone marrow, but for maturation they migrate to the thymus gland. So this name T, that is for T lymphocyte or T cells, that originated from the site of maturation, that is the thymus gland. So like the B lymphocytes, the T lymphocytes also express unique antigen binding molecule on its membrane or antigen binding receptor. It is called T cell receptor. Mature T cells will leave the thymus gland and they will migrate to other organs of the immune system such as the spleen, lymph node, bone marrow and the blood. T cell receptors only recognize antigen that are bound to cell membrane proteins known as major histocompatibility complex molecules or MHC molecules. So what does this say? The antigen must be bound to cell membrane proteins such as MHC molecule. Only then the T cell receptor can recognize this antigen. Okay. So there are two major classes of MHC molecules. Class 1 MHC molecule and class 2 MHC molecule. Class 1 MHC molecule is expressed by almost all nucleated cells of vertebrate species, and class 2 MHC molecule is expressed only by antigen presenting cells. Okay, antigen presenting cells such as macrophages, dendritic cells and all. So later on we will be studying how these macrophages are processing the antigen. Okay. So when a NEF T cell encounters an antigen combined with an MHC molecule on a cell, so we said earlier that if a T cell receptor has to recognize an antigen, that antigen must be bound to an MHC molecule, right? So when a NEF T cell encounters an antigen bound to MHC molecule on a cell, what happens? The T cell proliferates and differentiates into memory T cells and effector T cells. Okay. The T cells are classified into three categories: T helper cells, T cytotoxic cells and T suppressor cells. There are CD4 and CD8 membrane glycoproteins on the surface of T helper cell and T cytotoxic cell. So the T cells having CD4 membrane glycoprotein on its surface is called T helper cell. They will function as T helper cells, and the T cells having CD8 membrane glycoprotein on its surface will function as T cytotoxic cells. Okay. T helper cells recognize and interact with antigens that are presented on MHC class 2 molecule complex. Okay. So the antigen that is bound to class 2 MHC molecule, that will be recognized by T helper cells. And these T helper cells will get activated. And these activated T helper cells will become effector cells and they will secrete cytokines. And these cytokines will then activate B cells, T cytotoxic cells, macrophages and other immune cells. Okay. Now T cytotoxic cells, okay, T cytotoxic cells, they will recognize antigen that is bound to class 1 MHC molecule. So what we said about T helper cell recognize antigen bound to class 2 MHC molecule, right? So T cytotoxic cell recognize antigen bound to class 1 MHC molecule, okay? So then these T cytotoxic cell, it will proliferate and differentiate into effector cells known as cytotoxic T lymphocytes. So these cytotoxic T lymphocytes, they don't induce the secretion of cytokines. Instead, they will exhibit cell killing or cytotoxic activity. Okay. Now these T cytotoxic cells, they are mainly activated when a virus enters our body. So these cytotoxic T lymphocytes, they will eliminate any of the cells that display antigens such as tumor cells, cells infected with viruses and cells of a foreign tissue graft. Okay, so this is the structure of T helper cell and T cytotoxic cell. So on T helper cell, you can see there is T cell receptor and CD4 membrane glycoprotein on its surface. And on T cytotoxic cell, there is T cell receptor and CD8 membrane glycoprotein on its surface. So this is a virus infected cell and you can see there is class 1 MHC molecule on its surface. So the antigen, it is loaded onto this MHC1 molecule. Okay, antigen, it is bound to class 1 MHC molecule and it is presented to T cytotoxic cell. And the T cytotoxic cell will recognize this antigen MHC1 complex. Okay. And this is antigen presenting cell. Okay, antigen presenting cell such as macrophage. So this they are having class 1 MHC molecule and class 2 MHC molecule on its surface. And the antigen, it is bound to MHC2 molecule and it is presented to T helper cell. And T helper cell will recognize this antigen MHC2 complex. Natural killer cells are large granular lymphocytes. They don't express surface markers like B and T cell lineages. They display cytotoxic effect against wide range of tumor cells in the absence of any previous immunization with the tumor. These cells also play key role in host defense against tumor cells and cells infected with some viruses. Natural killer cells constitute 5 to 10% of lymphocytes in the human peripheral blood. So the natural killer cells express membrane receptors called CD16. CD16, they are receptors for the carboxyl terminal end of IgG molecule FC region. Okay, so these CD16 receptors, they will attach to the antibodies. Okay, they will attach to these antibodies and they will destroy the targeted cells by a mechanism known as antibody-dependent cell-mediated cytotoxicity. There are certain unique natural killer cells known as natural killer 1 T cells. They have combined characteristics of T lymphocytes and natural killer cells. Phagocytic cells. So, monocytes and macrophages are mononuclear phagocytic cells. Granulocyte monocyte progenitor cell differentiate into promonocyte and granulocyte, that is neutrophil, right? Promonocytes will then leave the bone marrow and they will enter into the bloodstream. And in the bloodstream, they will differentiate into mature monocytes. And these monocytes, they will circulate in the blood for about 8 hours. They will enlarge and then enter into tissues. And in the tissues, they will differentiate into specific macrophage and dendritic cells. Monocytes measure 2 to 15 μm with a single-lobed kidney-shaped nucleus. It accounts for 2 to 8% of blood leukocytes. Some of the functions of monocytes are they help in antigen processing and presentation, releases cytokines, they have specialized function in tissues, and another function is cytotoxicity, that is ability to destroy living cells. So this is the structure of monocyte and you can see it has a single-lobed kidney-shaped nucleus. Macrophages. So we said earlier that when the monocytes enter into tissues, they will differentiate into macrophages and dendritic cells, right? So this differentiation of monocytes into macrophages involves certain changes. That is, the cell enlarges 5 to 10-fold. Intracellular organelles increase in both number and complexity. Their phagocytic ability increases. What is phagocytosis? That is the ability of certain cells like macrophages to engulf and digest particles such as bacteria or debris. Okay? And they will produce higher level of hydrolytic enzymes. They will also secrete a variety of soluble factors. Free macrophages travel by amoeboid movement throughout the tissues. Macrophage-like cells serve different functions in different tissues and they are named according to their tissue locations. Okay? So there are alveolar macrophages in the lung, histiocytes in connective tissues, Kupffer cells in liver, mesangial cells in kidney, microglial cells in brain, osteoclast in bone. Activated macrophages express higher level of class 2 MHC molecule and function as antigen presenting cell. So we had already said that class 2 MHC molecule is found only on antigen presenting cells such as macrophages, right? So now we are going to describe how these macrophages process the antigen. Okay, once they engulf the antigen, how they are processing the antigen. So the macrophages on their cell surfaces have Fc receptors and these FC receptors will interact with the FC component of antibody IgG, and that will facilitate the ingestion of opsonized organisms. What does it mean by opsonized organisms? Opsonization means antibodies or molecules such as opsonins, they will attach to the surface of foreign particle or antigen, so that it can be easily recognized by these phagocytic cells such as macrophages. Okay, so in this case, antibody IgG has attached to the foreign particle or the antigen. So the FC receptors on the macrophage that will interact with the FC component of IgG, which is attached to this antigen, and that will facilitate the ingestion of the antigen. After ingestion, phagosome containing the microbe fuses with lysosome and they form phagolysosome. So phagosome, it is a vesicle and lysosome is an organelle. So they fuse together and form phagolysosome. And the microorganism inside the phagolysosome will be killed by reactive oxygen, reactive nitrogen compounds and lysosomal enzymes. Fragments of antigen are presented on the macrophage cell surface in conjunction with class II MHC protein for interaction with the T cell receptor of CD4 helper T cells. So what does it say? The fragments of the antigen that will be bounded or it will be loaded onto class II MHC protein. It will be bound with MHC2 molecule and then it is presented to T helper cells. This is followed by transportation of the complex to the cell surface by transporter proteins. Some of the other functions of macrophages are secretion of lymphokines, interleukin 1, interleukin 6, interleukin 12, tumor necrosis factor alpha to activate inflammatory response. Another function is secretion of granulocyte monocyte colony stimulating factors. So from the image you can understand the antigen is first ingested by the macrophage. So there is microbe in the phagosome and then there is lysosome. So the phagosome and lysosome, they will fuse together and form phagolysosome. Inside the phagolysosome, the microbe or the antigen, it is killed and fragmented. And then the fragment of antigen, it is bound to MHC2 molecule and the antigen is then recognized by T helper cell. Okay. So this is how the macrophages engulf the antigen and process it. So this is all about lymphocytes and phagocytic cells. I hope that the video was helpful for everyone. Thank you.